Hi, my name is Seth. I'm from Vanderbilt University Medical Center, and I'm here to tell you about our recent JCI paper. So this is an influenza virus. There's two major surface proteins on this virus. There's hemagglutinin, or HA, and this is the viral attachment factor. This is what mediates the interaction of this virus with sialic acid, which is on the surface of your cells. As a result, this protein can allow the virus to get into your cells, hijack the cellular machinery, and produce more copies of itself. There's another much less studied protein for influenza called neuraminidase, and in many ways, this kind of has the opposite function of HA. This is an enzyme that cleaves the sialic acid, and that allows newly formed viruses to leave infected cells and go on to infect other cells or other individuals. In our lab, we study antibody responses to both of these proteins in order to better understand the antibody responses people are making, as well as to understand how to design better vaccines or antibody therapeutics. So in this paper, what we did is we isolated antibodies from a single individual. To do this, we take B cells from their blood, sequence the genes that those B cells are expressing, and then express those genes as recombinant antibodies. When we did this, we found a really cool set of antibodies that were all genetically related, but these antibodies were able to effectively bind to N2 neuraminidase and inhibit it. And what we figured out is they're actually recognizing a particular part of the neuraminidase protein called the active site. This is the part of the protein that actually directly catalyzes this enzymatic reaction and then is responsible for cleaving sialic acid. So the way these antibodies are interacting is they're actually plugging up the Na active site. And then that is effectively inhibiting the virus and prevents the virus from getting off the surface of infected cells and spreading to other cells. And this is a pretty conserved part of this protein. So not only do these antibodies interact with N2, they also are able to inhibit N1 from both H1N1 and H5N1, which is an avian strain, as well as, excitingly, they can inhibit N9 neuraminidase from H7N9, which is another strain with pandemic potential. So by isolating these antibodies and the field isolating antibodies that are similar to this class of antibodies, we can better understand how these antibodies interact with the virus, how the virus might escape them, and how we can design more effective vaccines and antibody therapeutics.